and it's a shame we can't actually visit in person, but there's very good reasons why we can't. So what I would like to do over the next 40 minutes or so is to give a brief outline of our work to understand mechanotransduction and how it's been a journey to understand this protein in the center. I don't know if you can see my cursor actually. Here, this protein, this is Talin. This has been the focus of my work since 2006, since joining Dave Critchley's lab in Leicester. And over the last 15 years or so, we've really got a deeper understanding of what this molecule is doing and how it's coordinating way more than just integrin activation. And what I'd like to do is introduce how we've got to where we currently are. So the first part of my talk will be more data-based. And then the second part will be theory based on what I think that this is actually going on. So the title up until a few months ago was Deciphering the Talin Code. What I'd like to just finish on is a new theory which we're developing and testing, that these switches and this has the capacity to store information and to raise the idea, the notion that this might be a mechanical basis of memory with this MeshCODE theory which we've developed. And what I'd like to try and convince you is my feeling and my prediction is that memories in our brains are written into the synaptic scaffolds, and we'll see whether I can convince you of why we think that's the case. So this is a really old slide, which I've always had in since 2006. It shows a cell here spread, making these large adhesions where it's contacting this underlying extracellular matrix. And it's really old, but it really shows what we're looking at. These yellow linkages are the adhesions, and they tend to be coupled to the cytoskeleton shown here in red. So this cell was making lots of contacts. And what's really clear is that these adhesions aren't just holding this cell in place. These are providing key input into all of these different essential fundamental roles, from proliferation, contact inhibition, regulation of gene expression, development into tissues and organs. All of this is controlled, at least in part, by these integrin adhesion complexes. And these yellow cell matrix adhesions are mediated by this integrin family of cell adhesion molecules. And what got us particularly interested in the last couple of years, this regulation of gene expression. And it's probably shown best from this paper, which is now 15 years old, from Dennis Disher's lab. But this is just to show the power of this mechanotransduction, which we're working on, where what they do is a conceptually very simple experiment. They take some stem cells and they plate them onto different tissues. So in this experiment, everything is completely identical. It's the same cells, the same serum, the same extracellular matrix components. But the only thing that they vary is the elasticity of this underlying substrate. And purely by changing the physical environment, they can these cells will go from an undifferentiated stem cell type, and they will differentiate into lots of different lineages, driven purely by the mechanical connections that these cells make. So if you put it onto something really soft, it will make a fat cell or a neuron. If you put it onto more like glass-like substrates, you'll get more towards a bone level. But the only variable here is the stiffness. And that's different. And so what that leads to is this notion that these adhesions, these blue squares in this diagram, these are information processing centers, because that cell is feeling and experiencing its environment and then generating signals which change how the cell ultimately behaves to the level that you actually reprogram these cells. And to go into a little bit more detail of what these blue squares actually look like, this is the classic figure from Tony Kanchanawong and Claire Waterman over 10 years ago. But what it shows is, in this sort of sweetie diagram, is that these blue squares contain hundreds of these integrin receptors, and then the plasma membrane coupled to the actin cytoskeleton shown here in blue via this yellow and green bit. This is the linkage between the integrins and the actin, and this is where all of our research focuses, understanding this signaling layer comprised predominantly of talins and vinculins. And to go into a little bit more detail about what those blue squares look like, here's a cartoon of an integrin engaging to the extracellular matrix. And the integrins can switch between this low affinity state and this high affinity state where they actually engage. So the cell can grab on and let go in a very ordered fashion and coordinated fashion. And Talin, the protein which we work on, and you guys probably are familiar with already, is a cytoplasmic adapter. It binds to these short cytoplasmic tails of the integrin, and in doing so, it can coordinate the activation state of the integrins and determine whether the cell engages or lets go. But then Talin also makes those direct linkages between the integrin matrix complexes and the cytoskeleton within the cell. And this is really important. All of these linkages are essential for this to work. If you break any of them, this will fall apart. But what this means is that Talin, positioned between the integrin ECM on the outside world and the actin cytoskeleton, is perfectly positioned to sense and respond to forces, both external and internally. And this leads to this molecule being stretched. And that's where this notion of mechanosensitivity came from, that it can be stretched. And the classic example of Talin mechanosensitivity is the recruitment of the protein vinculin. And vinculin cross-links Talin to actin, reinforcing these adhesions, enabling them to go big and strong. And in essence, what we've got here is the core of pretty much all of these adhesive structures. You've got an integrin, a Talin, and an actin with vinculin, which comes in and reinforces them. But what's really striking is that onto that very simple core of 3 proteins, integrin, Talin, actin, you can assemble wildly diverse signaling unit complexes, and giving rise to these very different adhesive structures. So here's a fibroblast, but it could as easily be a metastatic cancer cell crawling along a surface. And here it's making its adhesive structures in green, and it's using them very dynamically to pull itself forwards. Those same complexes are shown here in a developing fly embryo, where they're much more long-lived, and they're actually involved in the myotendinous junctions, holding the musculature together. And then they make all of these weird and wonderful other adhesive structures onto this simple core. And what working hypothesis is that Talin adopts numerous conformational states which run different programs which tell the cell to do these different functions. And what we've got very interested in of late is these adhesive structures in the synapses of our brain. And these scaffolds, which I'll talk about a bit later, are essential for holding the integrity of each of these synapses together. They're ubiquitous in all of these. And again, there are Talin and integrin, Talin, actin linkages. So when we first came to this, I wasn't a mechanobiologist, I was a structural biologist. And what I was hired to do back in 2006 was to resolve this quandary, because this protein was known to be important, but it had 62 helices, but we didn't know what they looked like. And you can't get at the detail of what this molecule is doing with this level of resolution. So this slide summarizes about 4 or 5 years of work. And it leads to this, that what a Talin molecule looks like is it's got an integrin head at one end, a Talin head at one end, which binds to the integrin. It binds to actin at the C terminus, and then it's got 13 of these helical bundles arranged like this in this fashion. And one of the most useful things we did in this paper was to rename these to have a unified numbering system, R1 through R13. And these very boring looking domains are the focus of what we're most interested in the lab. And what we went on to then discover, not intentionally, but we found that each of these domains serves as a mechanical binary switch. And by that, what I mean is that this is a lower, this is the solution state. This is what these domains look like in the absence of any sort of input. They fold up into these helical bundles, but they can undergo these transitions in response to mechanical force, which causes them to transition from this closed conformation to this open conformation. And this is incredibly reversible. But what's striking is that held under force, both of these states are thermodynamically stable, which means that if you hold this molecule at either end, then these patterns of folded and unfolded domain will persist. And the reason we got into that was because our work on Talin R3, this R3 domain, and the discovery that this domain contains the high affinity binding site for a class of protein, a protein called REAM, which is a RAP1 effector. And this binds to this R3 domain in this folded form by this so-called helix addition mechanism, where this helix in green docks onto the side of this molecule. But what we already knew from previous studies in the lab was that this domain contains these high affinity vinculin binding sites, shown here in red. And I've only shown one here for clarity. But for vinculin to bind, it requires this domain to unfold, to expose this vinculin binding site helix to enable this high affinity interaction. So what that means is that this is what we've discovered, which we didn't appreciate for a bit when we first found this, is that this is a mechanical switch. In the low force condition, it binds to one set of proteins, the RAM, but then once you apply force to this, it transitions to this state and creates the high affinity vinculin binding site. So what that means is that force is driving a change in biological signaling. And I reckon that this is a textbook example of how you could convert a mechanical force into a biological signal by transitioning between two different conformations of the same protein to drive different responses. And this led to the collaboration, and as you're probably familiar with as well, with Yan Ji and his excellent postdoc Ming-Chi Yao in Singapore. And this was once we discovered this binary switch, this R3 domain, which could change between these two states. What we were able to do was we wanted to know how much force you need to apply to switch that switch to go from that state to that state. And the experiment, which you're probably familiar with, is to use these magnetic tweezers, where we can take a single molecule. Here we started with R1 to R3, 3 domains, covalently attach them to a cover slip or attach them to a cover slip, and then attach them to a magnetic bead. And that enables us to exert force on the bead, exert force on the molecule, and start to do biochemistry in the presence of mechanical force. And what this enabled us to do is when we pulled these 3 domains, what we see is that as we increase the force shown along this x-axis and measure just the extension in this experiment, then as you pull this molecule taut, it gets stretched, not surprisingly, but you see these large jumps in extension. And what these correspond to is to each of these domains unfolding completely independently. So this showed that each domain has got behaves as an independent switch, and each domain has its own unfolding threshold. So a different amount of force to trigger the transition of each of these 3 domains. And one of the most powerful combinations was to combine the structural biology and the biochemistry with the mechanobiology. And that enables us to make mutations which stabilize some of these domains. And we could show that we can change the force thresholds of these domains. So we can actually alter the switch patterns of these different domains. And if we stabilize R3, we see this big shift. So what that tells us is that R3 is the initial mechanosensing unit in Talin, and it requires about 4 or 5 piconewtons of force to trigger that transition. And also what you see here, this is 6 cycles of stretching and letting go, stretching and letting go. So in the absence of other factors, these domains will unfold and refold with really high fidelity. And that's what you want as a mechanosensor. Once you let go, you want it to reset itself back to its original state. And to summarize our understanding of what R3, this discovery of this switch in this R3, this is a key checkpoint in adhesion assembly. And I just wanted to go through that briefly now. So this switch, if we're looking at this switch between RAM in green and vinculin in red, then right at the very leading edge of the cell, we start to get enrichment of this RAM through RAT1 signaling. And what that means is that RAT1 recruits this RAT1 effector RAM to the plasma membrane at this very front of the cell. RAM's bound to Talin, so you start to bring all of these players to the front of the cell. If you start to imagine forces in this scenario, then the forces acting on Talin at these early stages are very low. But once Talin gets there, it can unfold, it can start to engage integrins, but this is still a low force environment. So all of the domains are folded. So this is held at the plasma membrane via the RAM RAT1 activity, the activation state of the RAT1. But what happens is you can get this key checkpoint where if Talin activates an integrin, which makes an attachment to the extracellular matrix and simultaneously couples it to the actin cytoskeleton, then all of a sudden Talin is under force now. Prior to that, this is a low force condition. But if Talin is attached to a firm surface at one end and then gets pulled internally at the other end, then you start to exert force on this molecule. The R3 domain unfolds first, and that releases the RAM, but simultaneously recruits vinculin. And then vinculin cross-links this whole unit together, making these adhesions big and strong, wrong way. And you end up with these very large vinculin-rich adhesive structures at the called focal adhesions. And this R3 switch is critical to this, because what it means is it stops these complexes forming at the wrong place in the wrong time. You'll only trigger this once you exert force onto these molecules. So having identified that R3 contained this key first switch, which triggered adhesion activation, we became interested in what this whole molecule looks like, and if all of these 13 domains have this switch-like behavior. And in particular, because our proteomics had found that there's over 40 different ligands which decorate this protein, this molecule. So expanding our single molecule approaches and stretching the whole of the Talin rod, then what we can find is we get this data, which is still jaw-dropping to us, because what this shows is the complexity of this mechanical signaling running through this an individual Talin molecule. And that's because each one of these steps in this trace correspond to one of these rod domains unfolding. So every single domain behaves independently as an independent mechanical switch. We've still got R3 down here, which triggers adhesion assembly, but then we start to get these steps. Each one of these is one of these switches unfolding. And because each of these domains binds to ligands in the folded and unfolded state, then every single one of these steps represents a change in the mechanosensing and a change in the biological signaling in response to force. And where this gets interesting is if we start to look at the type of ligands which are decorating the Talin rod, then there's all sorts of things which input into all of the different roles of the cell, really, from cytoskeletal regulators, transcription factors, proton pumps, kinases, microtubule regulators. All of these assemble onto these Talin molecules as a function of this switch pattern. So what we start to imagine is that these switches in Talin can be switched on and off to run different cellular programs, i.e. R3 switches on adhesion maturation. And one question we got asked early on was, this is all well and good, but if Talin's fully folded in a cell, then this would not be what you would see. But one of the things which you can see is if you look at Talin's extension in the cell, looking at end-to-end distances of Talin, then between 1 and 9 domains are unfolded at any one time. So between 1 and 9 of these switches are in the unfolded state. So it's conformationally very plastic. And we're still working on this. We don't actually know what all of these switches do. It's an ongoing area of research. There's lots of labs working on this. We kind of know that R3 is involved in adhesion maturation. If we stabilize that, we can disrupt mechanotransduction through these Talin linkages. What we showed a couple of years ago was that R7, through an interaction with a protein called CANC, directly controls microtubule targeting to adhesion sites. So this domain has to be in this folded form for microtubules to target to the focal adhesions. And this R8 domain directly couples these linkages to the cell cycle. And this is something which we published last month, but the CDK1, one of the major cell cycle regulators, sits on this domain and modifies these switches to change the signaling through these, change this mechanotransduction. So what we're trying to do, and what we're trying to develop a theoretical framework and then try and prove it as well, is to try and understand how this can occur, because this is frighteningly complex. You've got 13 switches on each of these Talins. You've probably got 100 Talins or more per adhesive structure. And you've also got multiple different cytoskeletal linkages. You've got the end-to-end, you've got all of the ones through vinculin, you've got a high affinity here to ABS2 to actin, you've got microtubules linking here. All of these inputs and all of these cytoskeletal linkages will exert force onto this system. And what we start to imagine is that those 13 domains will bind to different ligands as a function of these forces. And in a very low force condition, if all of these domains are folded, then you have all of these unfolded rod binders binding. But as you increase the force, you drive these transitions, and you're constantly changing and updating the signaling on that single molecule. And then as you exert more force, you cause another transition, changing that pattern of information and that signaling complex. But to be able to rationalize that and to consider all of these different forces, both from outside the cell and through all of these linkages, it's really complex. And we're not close to understanding this, but we think that there must be a code underpinning this. because we form with high fidelity, every cell knows what to do and it responds appropriately to the mechanical environment. So what we think is that Talin's forming different signaling platforms in these different scenarios, and depending on how much force in which direction, you assemble different signaling complexes. But somehow all of these competitive interactions, all of these different forces, all give rise to some form of meaningful response. And what that means is there must be a code to enable robust, reproducible signaling, and it must be metastable in response to all of these diverse multiple inputs. Somehow this all works out and works beautifully well. And that's what we're trying to understand in the lab. We're trying to understand this mechanotransduction code and how this can do all of these different functions through this same simple connection. And where this gets more complex is, on that previous slide, we're just considering what's going on with a single Talin molecule, which is already frighteningly complex. There's hundreds of Talins, there's hundreds of integrins, and there's many vinculins, plus the 250 other molecules which decorate this. So this is really, really complicated. This signaling machine which assembles, it's called integrin adhesion signaling, but it's much more complicated than that because you're assembling these complexes. So what we start to envisage is this meshwork of Talin and vinculin molecules, which cross-links and forms on the basal side of the cells, is a meshwork like code, which we dubbed the MeshCODE. This is a meshwork of switches, a vast array, and depending on the mechanical constraints and the signaling constraints, it becomes decorated with hundreds and hundreds of these different proteins, which we all know and love. Everyone's got their own favorite. And these decorate these complexes as a function of the patterns of the switches in the Talin molecule in a context-dependent manner. So what we can start to envisage is on the bottom of the cells, we've got this constantly sensory computational unit, which is constantly feeling the environment. And that's shown schematically here. And this provides constant mechanical signaling, feedback, and slight minor adjustments. And what we can start to imagine is what would happen if you applied a great force onto this meshwork. So some thought experiments of how this could function. And here's a cartoon of gravitational field waves, but it kind of shows the same thing. Is if you've got a cell and then you apply a force to it or a perturbation, then different size forces will have different size effects. But each one of these crosshairs would represent one of these Talin vinculin nodes. So as you provide a great amount of force, either intentionally or via damage or any different thing, then that will cause forces to be exerted highly specifically onto the adhesions at that site. And what will happen is they will experience forces. So at that site, you will start to change the mechanical signaling, and that will enable feedback. It'll either enable repair or it might enable the cell to differentiate or to change its behavior. So all of this can be ran through this, this MeshCODE. And so what will happen is you apply a force, it alters this coding, and then the cell adjusts as a function of that coding to change its behavior. And the cool thing about this is that this can all occur with just a handful of proteins. And even just considering in Talin and vinculin, there's 11 vinculin binding sites in Talin. Vinculin's a complex molecule itself. So just with those two proteins, and you'd imagine when you just talk about Talin and vinculin, just imagine these cross-links. But you can have incredible diversity and nuance in the signaling through these two proteins. And again, asking really simple questions is, could you imagine the scenario that each tissue has its own mechanical properties? And in doing so, that would create a bespoke MeshCODE, different patterns of switches folded and unfolded. I would say the answer is yes. I think the stiffness and the extracellular environment are different in all of our tissues. And secondly, could you imagine that each tissue has its own proteome, and that within that proteome, there's proteins that engage this Talin vinculin MeshCODE differently? And again, I'd say the answer to this is definitely yes. Each cell, each tissue has its own proteins being expressed. And that's sort of highlighted here. This is proteomics from different regions of the brain. Red, lots of protein, blue, reduced. But in each of the different, even in different slices of the brain, but not including the different organs, they're expressing different proteins and different amounts of them. So if they can engage this meshwork, which is also mechanically tuned to that tissue, then you can start to imagine that this Talin vinculin mesh code would enable incredible diversity and nuance in what's known as integrin signaling. So what I'm coming to believe, and what I'd like to argue today, is that this integrin signaling layer is actually a misnomer. This is a Talin signaling layer, or more specifically, a MeshCODE signaling layer. It's this meshwork of switches which is doing the signaling, and the integrin is actually just providing the attachment points and this feeling of the extracellular environment. So what I'd like to now talk about is a change in my thinking which occurred when I was at Sydney Airport waiting to catch my flight home, following a question I was asked. And it's to consider this is what I'd imagine my current view of what mechanotransduction looks like. You've got a cell which is crawling along a surface or under a microscope, and the mechanotransduction is what occurs from these switches in Talin and other mechanosensors. Other mechanosensors are available, but in Talin primarily here. And these switches, the pattern of these switches is going to be dependent on the cell's force generation machinery generating force against these, and also the extracellular forces. So depending on these two, you'll end up with a certain switch pattern which will instruct the cell how to behave. But what I suddenly did, I turned this on its head and started to think, what would happen if this is complicated and it's highly unpredictable because you've got two variables. If the extracellular environment was to become constant and predictable, and what I'm saying here is I'm not saying that this is never changing, because that can't ever be the case, but this is predictable. If the cell is maintaining its extracellular matrix so that it knows its stiffness, it knows its ligand density, if it's predictable, then here we've got a very different scenario. We've not got two inputs which are integrating into a response. Here what we've got is we've got a series of switches which are coordinated by the cell's force generation machinery against a solid, a predictable surface. And this suddenly means that these are known. This isn't a tug of war anymore. This is a cell which can operate these switches from within. And that's what I think can happen. I think the cell, and I think this happens all of our tissues, the extracellular matrix is tightly regulated and tightly controlled. So once the cell stops dividing and stops moving around, I think it fixes this and makes this a known. And this is what led to this paper, which came out a couple of months ago, which was in review for many, many months, because I had some quite robust discussions with some of the reviewers. But it's really interesting because it's really got it's stimulated a lot of thinking and a lot of very exciting conversations. But it led to this concept of this MeshCODE theory and what I think is the mechanical basis of memory. And I'd like to just talk about that now. And the fundamental concept is this, that these switches can be coordinated by the cell. The cell can pull on this and change the pattern of these switches in a known way. So one pull would change one switch, etc., etc. And so what we now start to think of is that Talin is actually a data molecule, and each of these switches is a binary switch. It's got the opportunity to be in a folded state or an unfolded state. But these can be switched back and forward by mechanical forces. And because this is held in a linkage, both of these folded and unfolded states are stable. So as long as you hold this under 3 or 4 piconewtons of force, then these domains remember the pattern that they're in. So we can imprint this with information mechanically. So so here's just 3 of these domains in series. In a very low force condition, they would all be in this folded 0, 0, 0 state, but a spike of contractility would transition one of these into an unfolded 1 state. And that would then change this information stored in this molecule from 000 to 010. And if you imagine that on a full molecule, again, just considering one Talin here for simplicity, then you've got 13 switches. When they make an adhesion, obviously some of these need to be in state, but you have a certain binary string of folded and unfolded. But if you apply contractility onto that adhesion by triggering RAC or cytoskeletal contractility, you can cause transitions of these domains, and in doing so, you can change the information stored in that molecule. And if you pull again, if that domain's already unfolded, it can't unfold again, so you can change the pattern further by unfolding another domain. And then you could have contractility decreases, which would provide the opportunity for domains to refold. So you could constantly be updating the information stored in each of these Talin molecules as a function of force. And so what I think and what I'm trying to argue here is that this is information that is being stored in these molecules. And this information is dependent on the prior events, because it depends what state this molecule is already in when you pull on it, and the future events. When you pull on it, that will update that information as a based on what the information already was, based on prior events. And we can show that this is an information molecule, both from our single molecule studies, but also from our simulations. One of the things which we've been able to do is we can precisely simulate now the folding and unfolding response of each Talin domain by measuring its kinetics. And what that means is that we can now start to do calculations on a single Talin molecule, and we can store this information persistently. And this is a simulation which we did where we're applying forces and then holding it and then letting it go. And we can store information on that individual Talin molecule. And we can even do sums on that molecule by how many contractility and how many decreases we exert. But all of this depends on the mechanical environment being well-defined and predictable. If that extracellular bit is changing or the cell's moving, it will still store information in these molecules, but they will be incredibly dynamic because there'll be these different variables. But if it's environment, if the environment is predictable, then this can be carried out with meticulous precision. And what we now, so the prediction from this theory is that the mechanical environment of the brain is perfect. And for a long time, mechanotransduction in the brain has been sort of not thought too much because of this very soft, this very low stiffness environment. But what that means is it gives great opportunity for each one of these synapses to form its own mechanical nest, in essence, of ECM, and maintain its own mechanical environment. And in doing so, that would enable mechanical computation across this linkage to be perfect. So I predict, and if you look at all of the papers on neuroscience papers, then during plasticity and during any sort of neuronal function, they're always updating and repairing and maintaining their ECM. It's to keep it in this mechanically predictable state to enable mechanical computation, is my theory. And if you look at what a synapse looks like, and this artwork's horrendous, I apologize, but this is a synapse. It's got the astrocyte, it's got the pre and the post-synaptic neuron. What you see is that these synapses are all scaffolded via integrin signaling, via these integrins and ECM and the actin cytoskeleton. So these switches which we've been talking about, this ability to store information in these molecules, in the shapes of these molecules which can store this information are holding these synapses together. So every single synapse has got this complex array of mechanical switches. And if you look at the literature, ECM has been well mapped out, integrins have been well mapped out, actin's been well mapped out, has been essential for all of this. But this has been very poorly studied, this mechanical linkage, whereas this scaffolding is what I think is the key to a lot of this. And that's because these switches, which can store information and change the signaling of that site, can be positioned around the periphery of all of these synapses. And to just try and conceptualize this a bit further, you can start to imagine this array of switches actually as an array of switches. And I've tried to show that schematically here with a Talin molecule of 13 switches coming down this side. And what you'd imagine is that each of these adhesive synaptic adhesions would have their switches in a certain pattern. But as you stimulate this synapse with electrical and chemical signaling, as is well mapped out, then the prediction would be that that would cause contractility of this cytoskeleton, and in doing so, alter the switch patterns of the talins, alter the data stored in that synapse, and in doing so, change the activity of that synapse if the prediction is that this is also involved in the, like integrin adhesion signaling is well mapped out in most cell types, but if it's also involved in the brain, then this would also alter the signaling of that individual synapse. And as a back of the envelope, if you imagine that there's 100 talins in each of these synapses, I suspect there's many more than that, and there's 8 functional switches per talin, then you might even start to predict that there's up to 100 bytes of binary information on either side of this synapse. Who knows? But that's all to be calculated. But the switches would coordinate this flow of information and store this information. So this leads to the theory which I'm presenting to you today of this MeshCODE hypothesis. And what I start to imagine is that this brain is a mechanical computer, and it's actually a computer which is running a binary coding. And these switches embedded into each of these synapses are the way that the organisms store and update information. And this transmission of this synaptic transmission would update the coding in both the pre and the post-synaptic side, and in the astrocytes and surrounding cells potentially, and it would update this information. But what's also quite interesting or nice to speculate is that these adhesive structures are essential for all of our tissues. Every single one of our cells, which isn't free floating, has integrin adhesions holding them in place. So you could imagine that this binary coding might be running in all of our tissues, updated, computed mainly in the brain, but then distributed throughout the whole organism. So this, if that was the case, if this is a binary coding, then it almost suggests, and it does suggest to me, and I'd like to argue that this is actually a machine code, which is coordinating the whole organism. And one of the reviewers was very keen to know how this coding would be read, and that's really important. And this is the Talin code re-envisaged in terms of binary information, that as you have a series of switches, only show 4 here for clarity, it will have a specific signaling output based on the ligands which it's bound to. So here we've got an O condition bound to a green, orange, and yellow ligand. So the signaling in that condition will be this. But then if you have synaptic signaling which causes contractility, if that does indeed convert one of these switches, then that synapse, those scaffold, will change and update its signaling, and it will now be O100, and it will be a green and now a blue and a yellow. And further contractility, you can't unfold that domain again. So you would unfold another domain and you would end up with, say, an O101. This is purely hypothetical, but as an idea, you'd be updating this switch pattern, and the readout would be the signals which are now assembled on that adhesive structure. And they'd be recruited and displaced from each Talin molecule at the adhesion site as a function of signaling. And what's well established outside of the brain is that adhesions are regulated by this contractility dependent growth. Forces exerted on an adhesion will cause it to grow and have stronger signaling. And if you don't stimulate that synapse, that adhesion, it will atrophy and slowly shrink again. So the readout, or one of the readouts of this coding, of this scaffolding, would be the relative redistribution of all of the ligands engaging the entire array of switches across the whole neuron. So these proteins would be moved around the neuron based on where there were binding sites to accept them. And the hypothesis would be that a stimulated adhesion would have a different pattern of folded and unfolded domains, different information, and the signaling through that adhesion would be different. And I don't want to spend too long on this because I'm probably getting towards my time, but just to conceptualize a few ideas and ground this in a neuroscience framework. And to do that, what I'm trying to show here is to try and show 4 synapses in 4 different states of activity. And this is a stimulated synapse. So the prediction from this theory is that a stimulation of a synapse, which causes vesicular release and signaling, will cause contractility in both the pre and the post-synaptic side. And you'll get a small burst of contractility and it will change these switch patterns. And at the presynaptic side, one possibility is that there's some part of the vesicular release which is dependent on the switch patterns in that site, or that this scaffolding is interacting with the switches in some way, so that the mechanical forces cause this release of the vesicle only at the stimulated site. So here you wouldn't get vesicles release because it's not stimulated, but this stimulation would cause vesicular release. Something which is really well mapped out in terms of it happens, but no one quite knows why it happens, is this concept of synaptic tagging and capture. And that's where a stimulated synapse will recruit molecules to it, and a stimulated synapse will grow and change compared to an unstimulated synapse. In the MeshCODE framework, in these switches, this becomes really easy to understand, because as you stimulate this synapse, if you unfold a domain, which then serves to recruit it to serve as a tag, which then recruits ligands only to that site, then only the stimulated synapse will have switches in that pattern, and they will recruit proteins to that site to cause the remodeling, whereas these ones won't be stimulated. And there's something called inverse synaptic tagging as well, which also fits nicely into this framework, where if you have dampeners of synaptic activity sitting on these unfolded domains, then you could imagine that as you stimulate this, if you unfold that, you might release these dampeners, and then these would then go and sit on the unpotentiated synapses where that domain was still in this folded state. So you'd be moving these soluble ligands around as a function of which synapses were being stimulated. So this one's activity would tune the activity of this one, etc. So they would all be talking through this mechanical coding, constantly turning them up and down and tuning their activity as a function of this mechanical signaling and this code, which this mesh code. And finally, you could imagine that a synapse which is stimulated multiple times would have multiple unfolding events and get strengthened. And each time you stimulate a talin, each time you pull on a talin, the change in the switches will be dependent on what state that molecule is already in. So you'll constantly update this coding, and then you'll unfold additional domains, which will only unfold if the domain which goes first has already gone. So you can constantly be updating this, and this might be what the memory trace is. This might be what an engram is, this pattern of information, which is both tuning what the status of each of the synapses, the activity running through them. And what this would mean is that you would have this scenario where this mechanical computation would be going on in all of these neurons, and they would constantly be turning up and down the activity of each of their synapses. And I'll just try and show it very, very schematically here. Here's a very simple reverberating circuit out of a textbook. But here I've just put one of these mesh codes underneath it. And at rest, you'd have a certain pattern of these, a certain activity of each of these synapses. But when you stimulate it, that would cause transmissions. And part of this return to homeostasis would involve synapses firing and updating this information in these different synapses, with the end product being that at the end, you would trigger the output, but you'd also find every single synapse's mechanical coding would have been updated as part of that process. And what that might mean is that this could either tune this such that the next time this happens instantaneously with much less calculation going on, or it might be updated every single time as a way to store and update information. And both of those could be rationalized in terms of this MeshCODE framework. And the theory, in the MeshCODE theory, the information stored in these synapses would also control the synaptic strength and probability of firing. That would be the theory which I'm trying to develop. And it also provides a nice way to understand how action potentials work. So these spike trains of action potentials where it's just an all or nothing, and the information is encoded in the frequency of these pulses, these spikes. This is in essence a binary coding, and it would work because each time one of these was firing, it would be updating the synaptic mesh coding, and it would constantly be sending signals both to the brain and then back. And at all these stages, these synapses would be getting updated with this new information, such that this, and it would be constantly updated and distributed throughout the animal. This hand would be primed, knowing it was already quite hot, and it'd be waiting for instructions from the calculation going on up here. But each of these synapses would already be updated with that information. So it would enable this to be incredibly fast, and it would also provide a basis for how these spike trains might be inputting information into this calculation by updating this binary coding. And what this enables me to start to think is that you could imagine that from birth, you'd have a certain pattern mesh code, and it would be constantly updated throughout the animal's life, all of the inputs and outputs and all of the calculations. So you'd be able to imagine an entire mathematical representation of the animal's unique life in this coding. So every single animal, depending on its lived experience, would develop differently and have different responses. So I'm going to wrap up, but just as a few thoughts, I think this provides a mechanical basis for how memories might be stored and how we might go about computation. And just as a final few thoughts, is that if you can imagine that every one of these mechanical switches can store one bit of information, then if you have an array of these switches, like you could store lots of information. Like when we first invented the transistor, whatever it was 150 years ago, 100 years ago, from one transistor to lots of transistors to trillions of transistors. And you start to store more and more information in these switches. And this leads to the second half of that MeshCODE paper, this one, it just came out last month, more philosophical, more thinking about this. But if you did have an array of these switches, these Talin, these adhesions, then you could store and write information to them. And what's really striking to me, at least, is that the cortex of our brain has an incredibly logical structure. It's got these millions of these cortical columns, really repeating, highly similar, with a very similar number of neurons. But if you look at these, each of these teardrops contains up to 60,000 different synapses. So this is an array of synapses. This is an array of MeshCODEs. With read-write going back from this trunk wiring to these. And if you compare this to what a solid state disk looks like, where you've got an array of binary switches, then I think that the brain, the cortex is like a solid state disk, where you've got this capacity to write information to these specific switches and store it in an ordered fashion in a specific place in memory. I think that that's what I would like to argue, that the brain memories might be stored in something analogous to a solid state disk. And if that was the case, then that would imply that each of these synapses can be addressable by neuronal activity, specifically addressable. I don't know whether that's known yet or whether it is. You sort of see these patterns, but you can't really, I think that this would suggest that everything happens and it's written to specific ones, synapses. But something which occurs is if you've got, and the calculations of how many Talins are, if you've got petabytes of data written into these disks, it's like if you try and find an old version of a file on your computer, it's really, really hard unless you've got a good ordering or a good search system. But you've got to have good memory allocation, writing data specifically to specific sites in a way that you can find it again. And that would suggest that you've got to have some form of addressable, the data has to be addressable. And I'll skip through this because this is a slide from a really nice review of a paper in Science a few years ago. But if you look at what happens during learning, then during the day, you get a lot of information. They've shown it conceptually here. And that's written into the hippocampus. But then overnight, that gets written into the cortex. And this, before you go to sleep, it's quite distributed and lots of information. But after you've been to sleep, it's written specifically to specific synapses. They don't necessarily view it as being written there, but the electrical fingerprint of that memory changes. And that would be in this MeshCODE model, that would be because those different synapses had been updated. And the REM sleep would be where this binary data is being physically moved around the disk. You'd be stimulating different circuits, updating this pattern of ones and O's stored in these synaptic adhesion MeshCODEs. So that's, that last 10 minutes was quite speculative, but that's just building and expanding from this theory. So just grounding it back on that this is all based on experimental data, and it's based on our research into how Talin functions, and this ability of it to have mechanical molecular memory, and this ability to store data persistently as a binary string. So each Talin can hold information, that information stored in molecules. We can work that out, that happens, and that's established. And so what that means is that scaling that up to consider these bigger pictures, and it scales beautifully to all levels of explanation, in my opinion, is that this is all based on well-established biophysical principles. And so what that means to me is that we've identified a physical location for data storage in cells. And it might represent a machine code. So all of our cells in our body are constantly being updated and kept in sync via updates to this coding. And the cool thing about this is that, as far as I can tell, is that this mechanical layer would integrate with what's really well established at the chemical and ionic signaling layers. And that's all well worked out, how the chemical and the impulses and the synaptic signaling occurs. This would sit underneath the hood, and it'd be constantly updating the information processing and storage. And it would be constantly tuning the activity of each of these synapses as a function of the signal, and tuning which synapse would fire next. And because this is identifies the conversion of storage of sensory and temporal inputs into a binary format, that to me identifies an addressable read-write memory system where you're constantly writing information and updating it. So I would like to finish by saying that, in my opinion, that this supports the view of the mind as an information processing system, or some form of organic supercomputer. And the key step for us now is to show that these switches and this molecular memory scales through the different levels of explanation, which is what we're currently trying to do in the lab. So I've probably gone massively over time. I apologize if I have, but I'd like to thank the people involved with this, in particular, Ji and Ming Chi, good long-term friends of mine, and very exciting. All the experiments we do, we find something completely novel out, and it changes your thinking completely. I've had lots of long philosophical discussions with Martin Schwartz and Nick Brown. That's been very useful. And the guys in the lab who are helping us to test and to prove this. And of course, the people who fund this work. And thank you for listening. And I'd like to take any questions if anyone has any.
Thank you so much, Ben. It's a fantastic talk. Like, it's really